This method generates and characterizes microparticle-dependent morphological structures of filamentous fungus Aspergillus niger to correlate fungal morphology with productivity. Cultivate a niger, with or without microparticles, in a 3-liter stirred tank bioreactor for 72 hours. To calculate specific productivity, take samples at defined time points and determine biomass dry weight and beta-fructofuranosidase activity. After 72 hours, examine the fungal morphology by microscopy and characterize by digital image analysis. Then combine relevant parameters of image analysis to a morphology number, which is mathematically correlated with the specific productivity. Ultimately, this method can precisely generate and comprehensively characterize morphology of Aspergillus niger for a better understanding of the morphogenesis of filamentous microorganisms.
Aspergillus niger is an important industrial working horse in biotechnology since many decades. One of the most intriguing and often uncontrollable characteristics of Aspergillus niger and related species is their complex morphology, ranging from dense spherical pellets to viscous mycelia, depending on culture conditions. The main advantage of our procedure is that, through addition of microparticles, we are now able to tailor-make fungal morphologies specifically for process needs. So far, no other method has been described for such a customization of fungal morphology.
This method can help answer key questions about the cultivation of filamentous microorganisms, because for industrial application it is crucial to control fungal morphology and also to distinguish between a well and a poorly producing fungal morphology. Our protocol provides the means for desirable customization and characterization of fungal morphology.